All right, welcome. Today we're diving deep into the microscopic underworld to unmask one of the most notorious families in the bacterial kingdom, Staphylococcus.
Yeah, I bet you have. That term gets thrown around all the time, right? From, a minor skin thing to really serious infections you can get in a hospital. But what is staph, really? Well, as it turns out, it's not just one bad guy. It's a whole family of them.
And this picture? It's the perfect family portrait. When scientists hit these guys with something called a Gram stain, they all pop up as these little purple spheres, or coccus, all clumped together. In fact, the name Staphylococcus comes from the Greek word staphylo, which means a bunch of grapes. Pretty fitting, huh?
So, before we single out the main troublemakers, let's get to know the whole family a bit better. Think of this as like the basic profile for any member of the Staph clan.
Okay, so we've got the purple grape thing down. They're also what we call catalase positive, which is just a simple lab test that separates them from another huge family of bacteria, the streptococci. And this is crucial, they're salt tolerant. That's a big reason why they're so comfortable just hanging out on our salty skin. So even with all these shared traits, there's a really clear dividing line in the Staph family. One single test that separates the most infamous member from all the rest. And this is pretty much the first question any lab detective is going to ask. And that test is called the coagulase test. See, this enzyme, coagulase, can actually make blood plasma clot. It's like the bacteria is building its own little fortress to hide from our immune system. So, if a staph species tests positive for this, we know we are dealing with the family's kingpin. If it's negative, we start looking at the other suspects.
And that coagulase-positive kingpin? That would be Staphylococcus aureus. The aureus part actually means gold because sometimes its colonies have this kind of golden color. This is the one you've definitely heard the most about.
Now, check this out. This slide shows a really cool visual trick that labs use to spot S. aureus. On the left, S. aureus is growing on this special plate. It's eating a sugar called mannitol and releasing acid, which turns the whole thing this bright, striking yellow. But look on the right. Most other staph species just can't do that, so the plate stays pink. It's such a clear, gotcha moment.
So, what makes S. aureus so nasty? Well, it comes loaded for bear. It has this thing called protein A, which is basically an invisibility cloak that stops our immune cells from grabbing it. It uses enzymes like coagulase to hide, and another one called hyaluronidase to just rip through our tissues and spread. And on top of all that, it pumps out all kinds of toxins that cause everything from food poisoning to toxic shock syndrome.
But the most terrifying weapon in its arsenal is its incredible ability to evolve and resist our antibiotics. That's what led to the creation of MRSA, methicillin-resistant Staphylococcus aureus, a superbug that has become this massive problem in hospitals all over the world.
Okay, next up in our lineup is the sneaky saboteur. Now this one is coagulase negative, but don't let that fool you for a second. Staphylococcus epidermidis is an absolute master of stealth and opportunity. The key to S. epidermidis's whole game is its superpower, creating biofilms. Just think of it as the slimy protective shield it builds all around itself. And once it's inside that fortress, it's incredibly hard for our immune system and even our antibiotics to get to it. And where does it just love to build these little fortresses? On any artificial surface we put inside the human body. We're talking IV catheters, prosthetic hips, artificial heart valves. Any medical device is basically prime real estate for S. epidermidis to move in and set up shop, causing these persistent, really hard-to-treat infections. And this creates a huge puzzle for doctors. You see, S. epidermidis is a totally normal resident of our skin. So when it shows up in a blood culture, they have to figure out, was this just a harmless skin bacteria that accidentally got in the sample? Or is it the actual cause of a dangerous device-related infection? It's a tough call.
Finally, let's meet our third family member, Staphylococcus saprophyticus. This one is also coagulase negative, but it has carved out a very specific and, let's be honest, very annoying little niche for itself.
This quote from our source material just sums it up perfectly. S. saprophyticus is a major cause of urinary tract infections, or UTIs, especially in young, sexually active women. So, unlike S. epidermidis, when this bug shows up in a urine sample, it's almost never just a contaminant. It's the culprit. Okay. So if both S. epidermidis and S. saprophyticus are coagulase negative, how in the world does the lab tell them apart? Well, they use another simple test, this time with an antibiotic disc called novobiocin. S. epidermidis is sensitive to it. the antibiotic kills it. But S. saprophyticus? It's just stubbornly resistant. That resistance is its calling card.
Okay, we've met our three main players: the superbug, the saboteur, and the specialist. Now, let's put it all together and see exactly how a lab detective would identify the culprit from this rogues gallery.
It's really just a beautiful logical flow. First, you confirm you're dealing with staph because of those grape-like clusters. Next, that all-important coagulase test. If it's positive, boom, case closed, it's S. aureus. If it's negative, you move on to the novobiocin test. If the bacteria is resistant, that points directly to S. saprophyticus, and if it's sensitive, you've identified S. epidermidis. Simple as that.
And this table right here, this is the ultimate cheat sheet. It brings everything we've talked about together. You can see the key tests: coagulase, novobiocin, and it even adds another clue called hemolysis, which is just the ability to break down red blood cells, something really only S. aureus does. The patterns are what matter. Coagulase positive, it has to be S. aureus. Coagulase negative with an infection on a medical device, you're suspecting S. epidermidis. And coagulase negative with a UTI, it is almost certainly S. saprophyticus.
Look, this isn't just some academic exercise. Being able to tell these bacteria apart is absolutely critical for taking care of patients. The drugs you use to treat MRSA are way different from the antibiotic you'd use for a simple UTI. Getting the ID right is step one in effective treatment, and it's a major battle in the larger war against antibiotic resistance.
Which, brings us to our final thought. As we come up with new drugs, bacteria like S. aureus are evolving new ways to fight back. It's this constant high-stakes arms race. As we get smarter at identifying them, they get smarter at hiding from us. It really makes you wonder, what's the next chapter in this ancient conflict going to look like?